I want you to try something for me right now. Just sit still for a second. Don't move a muscle. Don't even blink. Take a breath, let it out, and just exist.
Okay. Done. I am being a statue.
It feels peaceful, doesn't it? Maybe a little static. Like you are just this one solid thing, a single entity sitting there.
Sure. I feel stable. At rest.
Well, that feeling is probably the biggest lie your brain ever told you. Because if we had a camera, a magical camera that could zoom in past your skin, past the tissue, down to the absolute molecular level, what you would see is not peaceful.
Yeah.
It is absolute chaos.
I would call it controlled chaos, but yeah,
Yeah.
chaos is a good word for it.
It's a nonstop construction zone. Right now, as you're sitting there thinking you're doing nothing, you have something like 100 trillion cells functioning like 24-hour factories. I mean, they're burning fuel, building infrastructure, they're taking out the trash.
They're cloning themselves.
Yeah.
Constantly.
All with a level of precision that, frankly, our best human engineering hasn't even come close to touching.
Not even in the same ballpark.
Exactly. And that is what we are deep diving into today. We are looking at what is essentially the physiological manual of life. Specifically, we've got a pile of source material here covering Chapter 3, which is all about genetic control. And I'll be honest, when I first saw the topic list, transcription, translation, apoptosis, polypeptide chains, I thought, okay, this is going to be a dry biology class. I better get more coffee.
It happens. I get it. The terminology can be a real barrier to entry.
But then I started reading through the sources, and I realized I was completely wrong. This is not a biology class. It's like reading engineering code.
Yeah.
It's a story about a library that never, ever closes, a factory that literally builds itself, and a suicide switch that keeps you from turning into a monster.
That's a very dramatic way to put it, but you're not wrong at all. We are talking about the fundamental bridge between chemistry and life. How does a dead molecule in DNA is, chemically speaking, dead? How does it suddenly decide to become an eye or a liver or even a thought?
That is the mission for today. We're going to look at the library of instructions inside you, how those instructions are read, how the factory builds what you need, and this is the part that genuinely blew my mind. How that factory decides when to shut down, or even scarier, when it goes rogue.
Which is the core mechanism behind something like cancer.
Exactly. So let's start at the very beginning. The library. We all know the acronym, DNA, deoxyribonucleic acid. We see the double helix on TV. We know it's the blueprint. But physically, what are we actually looking at? If I could shrink down and hold a piece of DNA in my hand, what am I holding?
You know, it's funny. For all the amazing things it does, its chemical composition is surprisingly simple. You aren't seeing thousands of different exotic elements. You're basically looking at 3 core building blocks.
3.
That's it. First, you have phosphoric acid.
Okay, acid.
Second, you have a sugar, specifically a sugar called deoxyribose.
Acid, sugar, got it.
And third, you have a nitrogenous base. And there are only 4 types of these.
So, acid, sugar, and a base. That's the entire alphabet for life.
That's the whole kit. And they combine in a very specific way. 1 molecule of acid, 1 molecule of sugar, and one of the 4 bases link up to form a single unit called a nucleotide.
Okay, so the nucleotide is like the individual brick in the wall.
Perfect analogy. It's the brick. And if you look at the diagrams in the source material, like figure 3.2, you can see how these bricks stack up. The phosphoric acid and the sugar, they link together to form the long rails, the backbone of the structure.
Right, I'm looking at it now. It looks exactly like the side rails of a ladder, just twisted.
Exactly. Imagine a twisted ladder. That's your famous double helix. Those 2 long strands spiraling around each other are just alternating sugar and phosphate molecules. It's purely structural. It holds the whole thing together. But the actual information, the information isn't in the rails.
You don't read the binding of a book. You read the pages.
You read the pages.
Yeah.
And in this case, the pages, or the rungs of the ladder, are those nitrogenous bases.
And this is where the code lives.
This is where the code lives, and there are only 4 of them: adenine, guanine, thymine, and cytosine.
The famous A, G, T, and C.
Right. Now, looking at those diagrams again, you'll notice something really specific about the geometry. The rungs aren't just random. They fit together like perfect puzzle pieces.
It's not a free-for-all then. I can't just decide to hang out with G.
No, absolutely not. There's a very strict bonding rule. The source material calls it purines pairing with pyrimidines, which is the technical term. But simply put, adenine always pairs with thymine, and guanine always pairs with cytosine. A with T, G with C, period.
Okay, but why? Why is that pairing so rigid? Is it just a rule the cell follows, or is there a physical reason for it?
Oh, it's deeply physical. It
Mhm.
comes down to chemical stability and, honestly, just physical space. Imagine one type of base is long and the other is short. If you tried to pair two long ones, the latter would bulge out in the middle.
Right.
And if you paired two short ones, the rails would cave in. Adenine and thymine together just happen to make the perfect width for the rung. Same with guanine and cytosine. It keeps the whole structure uniform.
So it's literal architecture. It's about maintaining structural integrity.
It absolutely is. But there's a fascinating detail in the why here. The two strands of the helix, the two side rails, are held together by hydrogen bonds between these bases.
Hydrogen bonds. I vaguely remember those from chemistry class.
Well, here's the key question. Are they strong?
I I would assume so. You'd want your genetic code locked down tight, right? You don't want the pages falling out of the book.
And that's the trick. That's the genius of it. Relatively speaking, no, they're not strong. They are loose bonds. They're actually quite weak compared to the covalent bonds holding the sugar and phosphate backbone together.
Wait, loose, weak. That seems like a design flaw.
It would be if the book was never meant to be opened. But think about it. If those bonds were too strong, if they were like welding, you could never open the book to read it.
Mm-hmm.
As we're about to see, in order for the cell to read the code or to copy it to make a new cell, it has to physically unzip the DNA. It has to pull those two strands apart. If
Ah.
those rungs were welded together, life couldn't function. They need to be like Velcro, not superglue.
Okay, that makes perfect sense. So the backbone, the sugar and phosphate rails, that's the superglue. Don't break the spine of the book. But the code itself is Velcro, so we can gently peel it open, read a page, and then press it shut again.
Perfectly put. That's exactly the design principle.
Okay, so we've got this twisted ladder. We've got the four letters, A, G, T, C. How in the world does that spell make a human eye or digest the sandwich? It seems like a very, very limited alphabet.
Well, English only has 26 letters, and we managed to write Shakespeare. This code operates on a triplet system.
Triplets.
Right. The cell doesn't read one letter at a time, it reads three in a row. A sequence of three bases is called a codon. For example, if the DNA sequence reads GGC, that translates to the instruction, bring me the amino acid proline.
And if it reads, say, AGA.
That's the code for arginine. It's a language made entirely of three-letter words.
I see. And these words tell the cell which amino acids to grab.
Precisely. And amino acids are, of course, the building blocks of proteins. And proteins are, well, they're basically everything. So the structure of your cells, the girders and beams, they're the enzymes that catalyze reactions, the little machines doing all the work.
Now, the source material mentions something that seems like a contradiction. It says there are about 20,000 genes in the human genome, but we have over 100,000 distinct proteins. How does that math work? I thought it was one gene, one protein.
That is one of the biggest and most exciting realizations of modern genetics. We all used to think one gene, one protein. It was the central dogma for a long time. But it turns out the factory is much, much smarter than that.
How so?
The same segment of DNA, the same gene, can be spliced and diced in different ways to create different final messages. It's an incredibly efficient use of information. It's like having a single recipe for a cake, but if you choose to skip lines 3 and 5, you end up making a muffin instead. Same core ingredients, different outcome.
Wow. Okay, so this library, the DNA, is sitting in the nucleus. That the factory floor, where all the protein building action happens, that's outside the nucleus in the cytoplasm. The DNA is stuck, right? It's huge. It can't leave.
Exactly right. It's the master reference book in the library's special collections. It cannot be checked out, ever. It is strictly reference only. So if you need to build something out on the job site, you need to make a copy of the instructions.
You need a photocopy.
A photocopy. And this process of making the copy is called transcription.
Transcription, taking the notes.
Yes. And this introduces a major new player. A big enzyme called RNA polymerase.
I love the names of enzymes. They're so literal. Polymerase basically just means enzyme that makes polymers or chains.
Exactly. So here's the scene inside the nucleus. A signal comes in that a certain protein is needed. The DNA strand in that specific gene region unzips, but only temporarily. And only one of the two strands serves as the template.
So it doesn't copy both sides of the ladder.
No, just one. The RNA polymerase enzyme then jumps onto that strand. But it doesn't just start anywhere. It needs to know precisely where the recipe begins.
It'd have to look for a signpost, a start here sign.
It looks for a very specific sequence of DNA bases called a promoter. This is the code that effectively says, start reading here. Once the RNA polymerase latches onto that promoter, it begins its work. It starts to unwind the DNA helix, about 2 turns of the helix at a time, and separates the strands just enough to read them.
And as it moves along, it starts building a new strand, a new chain, but not of DNA. It's building RNA.
Correct. An RNA is the cheap photocopy. It's slightly different from the original DNA in a few key ways. First, the sugar in its backbone is different. It's ribose, not deoxyribose, hence ribonucleic acid.
Okay, makes sense.
Second, and this is a big one, RNA doesn't use the base thymine. It swaps it out for a different base called uracil. So wherever there was an adenine on the DNA template, the RNA polymerase puts a uracil opposite it instead of a thymine.
Now, I want to pause here because there is a detail in the source material about the energy of this process that really stuck with me. We think of reading or copying as a passive activity. I mean, I'm reading a book, I'm not really burning a ton of calories, but this process, the source implies it's almost explosive.
It is chemically aggressive.
Yeah.
And this is a key point to understanding life itself. Nothing happens for free. Every action has an energy cost. The RNA nucleotides, the individual building blocks waiting to be added to the chain, they aren't just floating around passively. They are activated.
Activated. What does that mean in plain English? How do you activate a molecule?
The cell attaches 2 extra phosphate radicals to each one. And these are derived from ATP, the cell's main energy currency. So these nucleotides are essentially triphosphates. They're loaded with energy.
Okay, so they're not just bricks, they're like spring-loaded bricks.
That's a great way to think about it. Imagine a spring compressed as tight as it can go and held down by a tiny latch. That's what those extra phosphate bonds are like. They're high-energy bonds just waiting to snap.
So every single letter waiting to be written into the new code is a loaded spring.
Exactly. When the RNA polymerase grabs a nucleotide and adds it to the growing chain, 2 of those phosphates break off. Snap. That high-energy bond is broken, and it releases a massive amount of energy, relatively speaking. That released energy is what physically powers the bonding of the new RNA chain. It forces the process forward with a lot of power.
So every letter added to the message costs a literal, tiny molecular explosion of energy.
A tiny explosion, yes. It's what drives the reaction and makes sure the bond is secure and the process moves in one direction. The cell is paying a very high price for every single bit of information it copies.
And the enzyme just keeps chugging along, adding base after base until it hits another specific code on the DNA.
The chain terminating sequence, the stop sign.
Right.
At that point, the enzyme just breaks away, the DNA zips itself back up like nothing ever happened, preserving the master copy, and the newly minted RNA strand is released into the nucleoplasm, the fluid inside the nucleus.
But wait, the source says what comes off the production line first isn't actually the final message. It's more like a rough draft.
Yes, that's a crucial editing step. This initial strand is called pre-mRNA. It's a raw tape. It contains the useful segments called exons. You can think X for expressed. But they're interspersed with these long, non-coding segments called introns.
Introns, intervening sequences. Is that basically just gibberish?
It's not necessarily gibberish. Some of it might be regulatory. Some of it might be old viral DNA from millions of years ago that got stuck in our genome. But for the purposes of this specific protein recipe, it's not needed. So before the RNA is allowed to leave the nucleus, it has to be edited. This is a process called splicing.
And there's a specific molecule that does the splicing.
Yep, a whole complex of them, actually, directed by something called small nuclear RNA, or snRNA. It directs the machinery to come in, snip out all the introns, and then stitch the remaining exons together.
It's literally like film editing. You cut out all the bad takes and the pauses and then tape the good scenes together to make the final movie.
And this goes right back to your earlier question about how 20,000 genes make 100,000 proteins. Depending on how you splice it, maybe you leave exon B in the final cut this time, but you cut it out next time, you can get totally different messages from the exact same gene.
That's how we get all that complexity from a limited library.
That's it. It's called alternative splicing. But once all that editing is done, you finally have your mature mRNA, messenger RNA. And that finished message is now ready to be shipped out of the nucleus to the factory floor.
Okay, so we're leaving the nucleus now. We are in the cytoplasm. The blueprint copy, the mRNA, has arrived. But a blueprint is useless without materials and workers. The source lists a whole cast of characters for RNA. We've met mRNA, the messenger. Who else is on the payroll for this job?
You have 2 other major types of RNA that are directly involved in production, and then a mysterious 4th type that acts as a manager. First up, you have transfer RNA or tRNA.
The source calls this one the carrier truck. I like that.
It's a perfect analogy. tRNA is a relatively small molecule, and it's folded into a kind of cloverleaf shape. Its one and only job is to go find a specific amino acid that's just floating around in the cytoplasm, pick it up, and transport it to the assembly line.
And it has to be incredibly specific, right? The truck that's designated for proline can't just decide to pick up arginine.
Extremely specific. Each type of tRNA is built to bind to only one of the 20 types of amino acids. And here is the absolute key to the whole process. How does the tRNA truck know where to drop off its payload? How does it read the blueprint?
It must have a code on it, too, a matching code.
It has an anticodon. Remember the three-letter codons on the mRNA message, like GGC for proline? Well, the tRNA that is carrying proline has the matching opposite code on its other end, CCG. It fits like a key in a lock. It forms those weak hydrogen bonds with the codon on the mRNA.
So the mRNA blueprint basically says, I need proline here, and the proline truck, the tRNA, sees that signal because its license plate matches, and it pulls in and docks.
Exactly. It's a physical key and lock mechanism. And the dock itself, the physical structure where all this happens, that's the third type of RNA. Ribosomal RNA, or rRNA.
The ribosome.
The ribosome is the machine. The rRNA, along with about 75 different proteins, folds up to create this massive molecular machine called the ribosome. It's the manufacturing plant. It provides the physical structure for the mRNA to thread through and for the tRNAs to dock into.
Before we turn the machine on, I want to ask about that fourth mysterious type you mentioned, the manager, microRNA.
Ah, yes,
Yeah.
miRNA, and its close cousin, siRNA, or small interfering RNA. This stuff is fascinating because for decades, scientists thought that a huge portion of our DNA was just junk because it didn't code for any proteins. We just ignored it.
Right.
But it turns out some of that so-called junk DNA is transcribed into these tiny little strips of RNA that don't build anything. They do the opposite. They stop things from being built.
They are the regulators, the quality control managers.
They are the silencers. The source describes a process where these miRNAs are first processed by a microprocessor complex in the nucleus, then by an enzyme called dicer in the cytoplasm, which are just great names, by the way.
The dicer. It sounds like a villain from a Gotham comic book. I am the dicer.
It really does. And then these little diced up RNAs assemble into a new machine called the RISC complex. And this RISC complex, armed with a miRNA, becomes a guided missile. It hunts down specific mRNA messages in the cytoplasm and binds to them.
And when it binds to the message, what happens?
It shuts them down. It can do it in two ways. It can either just physically block the ribosome from reading the message, like putting a piece of tape over the mouth of a speaker, or it can just signal for the mRNA to be destroyed entirely.
So if the cell's internal sensors say, whoa, we have way too much of this particular protein, shut down the production line, it sends out these miRNA hit squads.
Precisely. It's a fundamental mechanism for gene silencing. It ensures that genes that shouldn't be active, like a gene for making eye pigment in a liver cell, or just a gene that's overproducing and throwing things out of balance, stay quiet. It's an incredibly elegant form of quality control.
Incredible. Okay, let's finally run the assembly line. This is the big moment. Translation. We are translating the language of nucleic acids, the AUGC, into the language of proteins, of amino acids. The ribosome is the stage. The mRNA is the script.
The ribosome clamps onto the mRNA strand. It scans along until it finds the start codon. That tells it where to begin. Then it starts moving along the strand, reading it 3 letters at a time. Think of it like a tape head reading an old cassette tape.
Okay, so it moves to the first codon. A tRNA with a matching anticodon flies in and delivers its amino acid.
It drops off the amino acid. Then the ribosome shifts over to the next codon on the tape. The next correct tRNA flies in and docks right next to the first one. Now you have 2 amino acids sitting side by side.
And they need to be connected. They need to be welded together to start the chain.
Exactly. And an enzyme called peptidyl transferase, which is actually a physical part of the ribosome structure, acts like the welder. It forges a strong peptide bond between the 2 amino acids.
And I'm guessing this costs energy, too. We're back to the cost of doing business.
Oh, massive energy. The source says this is one of the single most energy-consuming processes in the entire cell. For every single amino acid that gets added to the protein chain, 4 high-energy phosphate bonds are split.
4, not 1.
4. 2 are used to activate the amino acid in the first place so the tRNA truck can even pick it up, and another 2 are used during the actual alignment and bonding steps on the ribosome.
That is just phenomenally expensive. When you think about the billions of proteins we're building every second, life is incredibly expensive from a metabolic standpoint.
It is. That's why you have to eat. That sandwich you digest is literally just fueling these tiny phosphate bonds to build the proteins that keep you alive. But the result is this growing polypeptide chain, a new protein that begins to snake its way out of the ribosome. And this happens fast. And often, to be super efficient, you'll have multiple ribosomes reading the same mRNA strand at the same time, one right after another.
The source calls those polyribosomes. It's like a convoy of factories all working on the same blueprint at once.
Efficiency is always the name of the game in biology. And once the ribosome hits the stop codon, the new protein is released. It travels into a structure called the endoplasmic reticulum. It folds into its specific 3D shape, and then it goes off to do its job, whether that's becoming part of the cell wall or becoming an enzyme to break down sugar.
So we've got the factory up and running.
Yeah.
But a factory without a manager is just chaos. How does the cell know what to build and when to build it? You touched on this with the liver cell versus the skin cell. They have the exact same library, the same DNA. Every single cell has the full manual to build the entire body. So how does a heart cell know to only read the heart chapter and to ignore all the others? This is all about regulation.
This is the critical difference between a pile of bricks and a cathedral. It's all in the organization. And regulation happens at two main levels: genetic regulation, which controls the library itself, and enzyme regulation, which is more about controlling the factory floor.
Okay, let's start with genetic regulation. This is about controlling access to the DNA.
Right. We talked about the promoter, the start sign for a gene. But it's not just a simple on-off switch. It's more like a complex dimmer switch, and it has multiple locks and keys.
The source mentions something called the TATA box.
TATA A. It's a common sequence of bases found in the promoter region. Special proteins called transcription factors have to bind there first to help the big RNA polymerase enzyme get started. But then you have other layers of control. You have things called enhancers. These are regions of DNA that might be thousands of bases away from the gene they control. But the DNA is flexible. It can loop around so that the enhancer region physically touches the transcription complex at the promoter, and when it does, it massively boosts the production.
It's like a turbo button.
It's exactly like a turbo.
Yeah.
And to keep things from getting out of hand, you also have insulators. These are chromosomal barriers that basically stop those enhancers from accidentally turning on the wrong neighboring gene. It's an unbelievably complex system of spatial organization.
And then there's the physical packing of the DNA itself, the histones. I found this absolutely fascinating.
This is one of the most fundamental levels of control. Your DNA isn't just a loose string floating in a bowl of nucleus soup. In each cell, you have about 6 feet of DNA packed into a nucleus that is completely invisible to the naked eye.
6 feet? How is that even possible?
To make it fit, it's wound incredibly tightly around these little protein spools called histones.
Like thread on a spool.
Exactly. Now, here's the key. If the thread is wound very, very tightly, if it's compacted, the RNA polymerase physically can't get in to read the gene. It's like super gluing the pages of the book together.
So to activate a gene, you have to physically unspool it from the histone.
Precisely. The cell has enzymes that can add or remove chemical tags to the histones, which causes them to either loosen their grip, exposing the DNA to be read, or tighten up, packing it away and silencing it. That's a huge part of how differentiation works. A skin cell has permanently packed away the genes for make insulin. They are boxed up in the attic and taped shut for good.
That makes so much sense. Okay, so that's controlling the library. Then you have enzyme regulation. This sounds more like real-time inventory control on the factory floor.
Right. This is all about feedback loops, simple supply and demand. If a cell needs to make a product, say purines, it uses a whole assembly line of enzymes to do it. But if the cell suddenly finds itself with a glut of purines, it's got too many, the purine molecules themselves will act as inhibitors. They will physically bind to the first enzyme in their own production line and shut it off.
It's like the product on the conveyor belt reaches the front and hits a switch that says, "We're full, stop the truck."
A perfect negative feedback loop. Conversely, you have activation. The source gives a great example with a molecule called KMP. When your cell's main energy molecule, ATP, is running low, a breakdown product called CANMP starts to build up. The CMP acts as a distress signal. It floats over and activates the enzymes that are responsible for splitting glycogen to release more glucose for fuel.
So low energy automatically triggers the release of more fuel.
Instantly. It doesn't need to send a message all the way back to the nucleus to make new proteins. It just turns on the machines that are already built and waiting.
Okay, let's zoom out again to the life cycle of the cell itself. We've been talking about the daily grind-making proteins, but eventually the factory needs to expand. It needs to clone itself. This is cell reproduction.
And before you can even think about splitting the cell in two, you have to split the library. You have to make a perfect copy of the entire DNA genome. This process is called replication.
The timing on this really interested me. The actual splitting of the cell, the process called mitosis, is actually pretty fast. The source says maybe 30 minutes, but the copying beforehand?
It takes hours, usually 4 to 8 hours. This all happens during a preparatory phase called interphase.
So how do you copy a molecule that is millions and millions of letters long inside a microscopic nucleus without getting it tangled up in a huge knot?
With our old friend DNA polymerase, a slightly different version than the one for RNA and a whole team of other support enzymes.
So we have an unzipper enzyme again.
Yep, an enzyme called DNA helicase comes in and creates a replication fork. It unwinds and splits the two strands apart. Now, here is where things get really tricky. And I need you to visualize this with me because there is a fundamental geometry problem here.
A geometry problem in biology.
Absolutely. DNA has a direction. For simplicity, we call the two ends of a strand the 5' and 3' ends. The DNA polymerase enzyme can only build in one direction. It can only add new nucleotides to the 3' end.
Okay, so it's a one-way street. The construction crew can only pave the road in one direction.
A perfect analogy.
Yeah.
But the two strands of the DNA helix run in opposite directions to each other. One runs north, the other runs south.
Oh, okay, I see the problem. So if the DNA double helix is a two-lane highway, you've got one lane of traffic going north and the other lane going south.
Exactly.
And the DNA polymerase is a paving machine that can only drive north. So on the northbound lane, which we call the leading strand, it's easy. It just gets on the road and zooms along, laying down new pavement as it goes. It's one smooth, continuous process.
Continuous replication, that's exactly right.
But what about the other lane, the lagging strand? That road is heading south, but the paving machine can still only drive north. So how does it possibly copy a southbound road while it's physically facing north?
It has to do a backstitch.
A backstitch.
Think about it this way. The machine drives a little bit up the road, north. Then it has to jump backwards down the newly unzipped strand and pave another short northbound segment. Then it waits for the helicase to unzip more of the road, jumps backwards again, and paves another little northbound patch. It's working in reverse, but in these tiny disconnected chunks.
Wow, that sounds incredibly inefficient and complicated.
It is complex.
Oh, yeah.
And it creates these little islands of newly copied DNA that are called Okazaki fragments.
Named after the Japanese researchers Reiji and Tsuneko Okazaki who discovered them.
The very same. And because you now have all these disconnected fragments, these potholes in the new road, you need another specialized crew to come in. An enzyme called DNA ligase acts as the road crew to come in and stitch all those fragments together into one smooth, continuous highway.
So one side of the fork is this beautiful, smooth pour of concrete, and the other side is a frantic mess of a bunch of patched sections being stitched together just in time.
That's a pretty accurate picture of what's happening. And this whole lagging strand issue isn't just a weird quirk, it's actually a huge deal for aging and the lifespan of a cell, which we'll get to. But first, just consider the potential for errors. With all this backstitching and jumping and patching, you'd think the copy would be full of typos.
Yeah, that sounds incredibly prone to error.
It would be. But the actual error rate is astonishingly low. And the reason is proofreading.
The polymerase actually checks its own work.
It does. It has a built-in delete key. As it adds a new base, it checks to see if it's the right fit. If it accidentally places an A where a G should be, it recognizes the physical mismatch. It pauses. It acts like an exonuclease to cut out the wrong nucleotide. It puts in the right one, and only then does it move on.
So it's literally typing, looking back over its shoulder, hitting backspace to fix a typo, and then continuing to type.
Every single time. This incredible proofreading function keeps the mutation rate down to about 1 in a billion. But once all 6 feet of DNA are copied, we are finally ready for the main event. Mitosis. The physical splitting of the cell.
This is the part that's just visual drama. If you look at figure 314 in the source material, it's like a choreographed ballet.
It really is. The whole process starts with prophase. The chromosomes, which are usually loose and spaghetti-like so their genes can be read, now coil up super tight. They condense into those classic X shapes we see in textbooks. This is like packing up all the furniture in the house before a big move.
And then the centrioles move to opposite sides of the cell.
Right. These are little structures that act like anchors for the whole operation. They move to the opposite poles of the cell, north and south, and they start shooting out these protein filaments called microtubules. This forms the spindle.
Next is permetaphase. The walls of the library, the nuclear envelope, just dissolve.
And those microtubules, those ropes, attach to the center of each chromosome at a point called the centromere. It's like a team of movers throwing grappling hooks onto each box of furniture.
Then comes metaphase, the alignment.
The molecular motors on the microtubules start pulling and pushing the chromosomes until they are all perfectly lined up right in the middle of the cell on what's called the equatorial plate.
It's that moment of perfect tension right before the snap. And then anaphase.
The separation. The two identical halves of each chromosome, the chromatids, are violently ripped apart. The motors reel them in, pulling the complete sets of genetic information to opposite sides of the cell.
And finally, telophase, the cleanup and the split.
Exactly. New nuclear membranes form around the two new piles of DNA at each end. And then the cell itself physically pinches in the middle.
The source mentions a contractile ring made of actin and myosin.
The very same proteins that are in your muscles, they form a belt around the middle of the cell and just tighten and tighten till it snaps, creating two new identical daughter cells.
And now we have two factories where there was only one. But this leads to a huge philosophical question. If every time a cell divides, it's just making a perfect clone of itself, how do we grow up? How does a single fertilized egg turn into a complex human with eyes and a liver and skin?
That is the magic of differentiation.
The source material brings up a famous experiment with a frog that I think explains this concept perfectly.
Yes, this was a landmark experiment. It proved a vital concept. Researchers took the nucleus from a highly specialized intestinal cell of a tadpole, a gut cell, and they implanted it into a frog egg that had had its own nucleus removed.
So they put a gut cell instruction manual inside a blank egg cell.
Right. Now, if the gut cell had somehow lost all the genes to be anything else, you'd expect it to just grow into a pile of gut cells. But that's not what happened. It grew into a completely normal frog.
Wow. Which proves that the intestinal cell didn't lose the genes to make an eye or a leg. It just had them turned off.
They were repressed. That's the key.
Yeah.
All the potential is always there in every cell.
Hmm.
Differentiation is just the process of selectively and permanently repressing different parts of the genome. It's all about which chapters of the book are glued shut forever.
But the factor doesn't run forever. We age. The system degrades over time.
It does. And do you remember that lagging strand problem we talked about during replication? The backstitching?
The northbound paving machine trying to work on the southbound lane.
Right. Well, because of the way the initial RNA primer has to sit on the very end of the strand to get the copy started, the polymerase can never, ever copy the absolute tip of the DNA tail.
It essentially runs out of road before it gets to the end.
It runs out of road. So every single time a cell divides, the DNA in the daughter cells gets a tiny bit shorter.
That sounds incredibly dangerous. You're losing lines of code with every copy.
You would be, except that nature has a built-in buffer system. They're called telomeres.
The protective caps on the ends of the chromosomes.
Think of the little plastic aglet at the end of a shoelace that keeps it from fraying. Telomeres are just long, repetitive sequences of nucleotides at the ends of the chromosomes. TTG, TTG, over and over. They don't code for anything important. They're just disposable buffer.
So with each division, you lose a little slice of the telomere instead of a slice of an important gene.
Exactly. But the telomeres aren't infinitely long. So after enough divisions, maybe 50 or 60 times, the telomeres are basically gone. Then you start cutting into the actual genes themselves. The shoelace starts to fray, the cell becomes senescent, it gets old and stops dividing, or it just dies.
Unless you have an enzyme called telomerase?
The enzyme that can rebuild the caps. Certain cells have it, like stem cells, because they need to be able to divide for a very long time. But most of your normal cells do not. And unfortunately, cancer cells often figure out how to turn it back on, which gives them a form of immortality. For most cells, though, telomere shortening is a built-in lifespan, a ticking clock.
Speaking of death, the source makes a clear distinction that there's a right way and a wrong way for a cell to die.
Very different. Necrosis is the wrong way. That's death from injury, a burn, a cut. The cell swells up, it bursts, and it spills all its guts and inflammatory chemicals everywhere. It's a messy, damaging process. It's a murder scene.
But there's a clean, quiet way. Apoptosis.
Programmed cell death. It's the cell's suicide pill. If a cell is damaged beyond repair, or if it's infected with a virus, or if it's just old and not needed anymore, it can trigger an internal suicide cascade.
And this involves a family of enzymes called caspases.
Exactly. A family of proteases, protein-eating enzymes. They get activated inside the cell. They start chopping up the cell's proteins, the cytoskeleton collapses, the DNA gets fragmented, the cell shrinks to a neat little ball, and its surface changes to send out an eat me signal so that an immune cell called a macrophage comes along and just gobbles it up.
It's tidy. No mess, no inflammation.
It's absolutely vital for life. Without apoptosis, you would have been born with webbed fingers and toes. The cells between them die off in the womb to separate them. Your brain would be dangerously overcrowded with unneeded neurons. You'd be riddled with damaged, potentially cancerous cells.
But sometimes the police fail. Sometimes the DNA damage isn't fixed and the cell doesn't commit suicide. Sometimes it mutates and it just keeps growing.
And that is the beginning of cancer.
We spent almost an hour talking about how brilliant and intricate this system is. The proofreading, the repression, the histones, the suicide switches. I mean, it sounds foolproof. But then we had the devastating statistic that so many people get cancer. If the system is this smart, why does it fail so often?
Because it's a numbers game.
Yeah.
And the sheer numbers are stacked against you. You have roughly 100 trillion cells, and many of them are dividing constantly. Even if the error rate is only 1 in a billion, that's still thousands and thousands of mistakes happening in your body every single day.
So you're saying right now, as we're talking, I have cells in my body that are making mistakes and trying to turn into cancer.
Almost certainly. But in a healthy person, the police, the DNA repair enzymes, and the apoptosis pathways are catching them. The suicide switch is being flipped. But cancer is what happens when a cell, through a series of unlucky mutations, evolves its way past all the police checkpoints.
The source breaks down the cause. It's always mutation.
Always. But it's almost never just one mutation. It's a series of hits. You need to take out multiple safety systems.
And it describes two main types of genes involved: proto-oncogenes and anti-oncogenes.
Right. Think of proto-oncogenes as the gas pedal for cell growth and division. They're normal, healthy genes that say go. If they get mutated into what we call oncogenes, it's like the gas pedal gets stuck to the floor. The car just accelerates uncontrollably.
And anti-oncogenes.
Those are the brakes. They're also called tumor suppressor genes. Their job is to stop the cell from dividing if something is wrong. They're the checkpoint cards. If these genes get inactivated by a mutation, you have no brakes.
So a stuck gas pedal and cut brake lines.
A recipe for disaster. And the things that cause these mutations, the source lists the usual culprits. Ionizing radiation like X-rays or gamma rays that can physically shatter the DNA strands. Chemical carcinogens like the dozens of them found in cigarette smoke or certain industrial dyes. Even chronic physical irritation. And of course, certain viruses that can actually insert their own DNA right into your chromosomes, scrambling your code.
And once a cell becomes fully cancerous, it behaves differently. It's not just growing fast. The source says it's almost evil. It's parasitic.
The source highlights a few key terrifying traits. First, a lack of adhesiveness. Normal cells have a social contract. They like to stick together in their proper tissues. If a liver cell touches another liver cell, it gets a signal to stop growing. It's called contact inhibition.
It's like, okay, I'm touching my neighbor. I shouldn't push any further.
Right. Cancer cells lose this. They become slippery. They don't stick well to their neighbors. And that's a key reason why they wander off the process of metastasis. They can break away, drift through the blood or lymph, and set up a new shop somewhere else in the body.
And then there's the scariest part, angiogenesis.
This is perhaps the most diabolical part of it all. A growing tumor is hungry. It needs a massive supply of food and oxygen. So it starts releasing chemical signals, essentially hacking the body's normal infrastructure request system, screaming, "Send blood here, build a road to me."
So it actually tricks your own body into building a brand new highway of blood vessels right to the tumor's front door.
Exactly. It forces your body to build the infrastructure to feed the very thing that is killing it. And because cancer cells are so metabolically aggressive, they steal all the nutrients from the surrounding healthy cells. This is why cancer patients often experience that severe wasting away. The tumor is literally starving the rest of the body to fuel its own unlimited selfish growth.
It's a very dark turn for our factory tour, but I think it highlights just how incredibly precise and balanced the normal system is.
That is the ultimate takeaway. When you really look at how complex the regulation is, the promoters, the histones, the murinase silencers, the proofreading, the apoptosis checkpoints, the real miracle isn't the cancer happens, it's that it doesn't happen more often.
Right, billions of cells dividing, each one copying 6 feet of code and getting it right almost every single time.
It is the most sophisticated piece of machinery in the known universe. And it's running inside you right now without you having to lift a finger or even think about it.
So let's try to wrap this up. We started with a simple chemical acid, sugar, base. We built a helix. We learned how to read it, how to transcribe it into RNA using those tiny explosive phosphate bonds, and how the ribosome factory translates that message to build proteins.
We saw how the cell regulates itself, turning genes on and off like light switches, using everything from physical packing to tiny RNA hit squads. We saw how it copies itself with that bizarre leading and lagging strand backstitch. And we saw the constant, delicate balance between life replication and death apoptosis.
It really does change how you see yourself. You aren't just one person. You are a walking, talking colony of 100 trillion distinct living entities, all working in concert.
And the health of the you depends entirely on the fidelity of that ancient code and the incredible regulation of those tiny factories.
Here's my final thought for you to chew on, something that wasn't really in the text, but that this all leads to. We've spent this entire deep dive talking about how mistakes, mutations are the enemy. They are the causes of cancer and disease. But without mistakes, without that tiny fraction of error in the code, repeated over millions and billions of years, would we have evolution? Would we even be here to talk about it?
That is the ultimate paradox of life, isn't it?
Yeah.
Perfection means stasis. If the DNA polymerase had never made a single mistake, we'd probably still be single-celled organisms floating in the primordial soup. Error means risk, but it also means possibility.
It's a very, very fine line between a monster and a miracle.
It certainly is.
Thanks for diving deep with us into the blueprint of life.
It was a pleasure.